So I'll turn it over to Jim, who's going to introduce our speaker.
All right. Well, tremendous pleasure to be able to introduce Dr. Jonathan Losos today. Many of you know that this is Jonathan's second talk that he's given for us. He gave Herp Group seminar on Monday night. It was a spectacular presentation. And before that presentation, I gave him sort of a very brief introduction. And so even though I know I'm going to eat into your time a little bit here, I'm going to give you a better introduction for your NBC lunch seminar. And it's really easy to give a tremendous introduction for Jonathan because he's done so much. So just a little bit, his bio. So he graduated with his bachelor's degree from Harvard University, and then he came here for his PhD, and he was an MBZ graduate student, and officially a zoology graduate student. He got his PhD here in 1989, and then he had a couple of years of postdoc at UC Davis. Were you in Tom Shaner's lab?
More or less.
More or less. At Davis. And then he started his first faculty position at Washington University in St. Louis, where he built a really incredible program that produced lots of really high-quality graduate students and postdocs who came out of that department. And it was one of the top programs for herpetology for sure in those days. But Harvard, as they do, recognized his greatness and recruited him, so that he could go home again to his undergrad institution. And so he's been a professor at Harvard. He went to Harvard in 2006, and he's been there ever since. And he's a professor there in the Department of Organismic and Evolutionary Biology, and then also a curator in the Museum of Comparative Zoology. So Jonathan has won lots of awards, and he started winning awards early, and he's continued to win awards. And so I want to list some of those awards. It's not all of them. The list is too long, actually. So in 1991, he won the Dobzhansky Prize. And then in 1998, he won the David Starr Jordan Prize. He had a John Simon Guggenheim Fellowship in 2005. He was the recipient of the E.O. Wilson Naturalist Award from the American Society of Naturalists in 2009. He recently won the David Gerard Elliott Medal of the National Academy of Sciences. That was in 2012, which is probably, well, I'm not going to say it. Maybe it's the preamble to getting into the National Academy of Sciences, probably your next step. And then in 2012, he was elected to the American Academy of Arts and Sciences. So that's just a subset of the many awards that he's won. Jonathan has an incredible publication record. I tried to count the publications. You should number them on your website. I might have lost count. It was at least 180 publications, and that doesn't include the 5 books that he's written, plus the 8 textbooks that he's either authored or co-authored. It's incredible, and he has. This is probably the highest praise that I can give somebody. He has a David Wake-esque record of graduate student and postdoctoral mentorship. If you ever want to just be, if you're a professor, if you want to be humbled, or if you're anybody who wants to be impressed, go look at the list of Dave Wake postdocs and graduate students and where they are now. Very few people can compare, but Jonathan can. He's had 21 postdocs that came through his lab that have already left, many of whom are in faculty positions, as well as 17 graduated PhD students, if I count it correctly. Many of these people, if they're not already in faculty positions, they will soon be in faculty positions. So it's a really incredible, record of mentorship. So, with all these accomplishments, you can imagine that Jonathan might be too busy to talk to graduate students and that sort of thing. But one of my great memories of being a grad student was meeting Jonathan. I think when I was a master's student, before I was even a PhD student, meeting him at meetings and him taking the time to talk to me. And I think that for many people in the room, you probably met Jonathan. If you're young, like me, I'm not very young. Remember Jonathan? Maybe you met him like I did while you were still a graduate student. I think that's one of the really great features of Jonathan's personality is that he's willing to talk to people at all levels. And so I just want to thank you for that. And I'm going to give you the floor now before I eat up too much more of your time. It's just a terrific pleasure to have Jonathan here to speak to us. One of the nice people.
Yeah. Lights. Somebody, lights back here.
No, other side.
Well, thanks, Jim. That was just a wonderful introduction. Thank you so much. I promised my mom I would do this. Good enough. Good enough. So, this is just an incredible thrill for me to be back here giving my first museum lunch in, I think, 26 years. And so it's taken me a walk down memory lane. Museum lunch back when I was a graduate student here in the 80s was very important to graduate students. We not only had wonderful talks, but we got to see the interaction with the great questions and so on, and got to see what it was like really to be a colleague. And then we gave our own talks, and it was very important. I can't remember how many talks I gave but I remember one very clearly for the questions I got, and I can tell you about that later. But anyway, it's great to be back here. And in fact, in thinking about this, I've decided to give this talk very much oriented towards the students and younger scientists here. And I hope you older scientists don't get annoyed by me having the conceit that I have something to say to young scientists. But anyway, that's how I've organized this talk. And particularly in thinking about some of the younger scientists here, some of the students in the museum and elsewhere, there are some traditions that I think are at risk of being lost. And I think it's important to remind you of what has happened in these hallowed halls. I refer, of course, to tie day. We used to have a day where everyone would wear a tie. And I mean everyone. And I strongly urge you to bring tie day back. There was also hat day. And most people wearing this one hat, but some people had to do it differently. Actually, there was no hat day. I just bought a very cheap hat on Telegraph, and look at all these professors who will put on a hat. I think it says something about Berkeley faculty. So anyway, it's the other thing about my time here is I was thinking back to the other students who were students along with me here in the museum or in at that time the zoology and botany departments. And it's a pretty incredible bunch of people. You look what they've accomplished today, and just people, Barry Sonervo, Mark Kirkpatrick, Kisa Nishikawa, and many others. The people with whom I was students and who are postdocs are now leaders in the field. And from my interaction with the students here today, you guys are equally as talented as we were back then. And so I guess my point is, look around you, and these are the people who 20 years from now will be leading the field. And Berkeley has been and continues to be just an incredible place to produce the people who are leading science. And so you all are very fortunate to be here. One person, however, who did not get his degree here is Charles Darwin. And I know it's kind of hokey to start a talk with a Darwin picture. Everyone does that. But I think I have a slightly different twist on why I'm mentioning Darwin. And that is an aspect of Darwin's career that doesn't get so much attention. And that is Darwin the experimentalist. Now, it's not widely recognized by many people, but Darwin, back in the 1850s, had an experimental mindset, well before that was standard in science. And he had his ideas, and he would devise experimental ways of testing them. For example, he was interested in how is it that plants make their way to oceans, and could seeds survive transit in salt water? And so he put a bunch of seeds in salt water and found out. Or he was interested in how plants grow towards sunlight, and so he did manipulations on plants to find out what part of the plant was important for it moving towards sunlight. Or he did experiments on earthworms, and of course he was the one who figured out how the soil was created by earthworms. But he was also interested, could earthworms actually respond to sound? And so he played music to them to see what would happen. This was recreated in a Harvard class recently. So so Darwin had this experimental mindset, and yet his greatest idea, evolution by natural selection, he never tried to test experimentally, which seems a little bit odd, but of course we know the reason why. And that is, he was under the opinion that evolution proceeded so incredibly slowly that you couldn't possibly observe it over any span of a human lifetime. And so it never would have occurred to him to do an experiment to test his idea. Now, we know that, and of course his ideas about the pace of evolution were not informed by any data, because no one was studying that at the time. It was basically his own intuition, was borrowing common ideas from geology at the time, and Victorian sensibilities about the proper pace of change in the modern world. We know, of course, now that Darwin didn't know two things, that he was wrong about them. One was, of course, heredity. How could he have known about how heredity worked? But the other thing that he didn't know was that evolutionary change can occur very rapidly, particularly when natural selection is strong. And we now know that Darwin was wrong about this. We first learned this from laboratory population studies starting in the 20s with fruit flies, where if you select on a fruit fly for almost any trait, it will evolve very quickly. Then in the 1950s, the Vista and Vecillaria, seeing the change, or documentation of the change from the peppered morph to the black morph, changing very quickly. And then, of course, all the animals and plants adapting to our pesticides and insecticides, and the bacteria adapting to our drugs and so on. We've seen rapid change all around us. And I think the key development in this work, all these examples I've just given you were all human modifications or in the lab. But when we saw that it occurred in the natural, seemingly unaffected part of the world, unaffected by humans, it became clear that evolution could occur very rapidly. I'm referring, of course, to the work on Darwin's finches by Peter and Rosemary Grant, who showed that the environment can change greatly from one year to the next, and when it does, natural selection, measured here, could change greatly, and as a result, that evolution can occur very rapidly. The point of all this is it's now become very clear that in this way, Darwin was absolutely wrong, that in fact evolutionary change can occur very rapidly. And in fact, it's become, in just a very short period of time, it's gone from something that we thought didn't happen to the expectation. Now people assume that populations will evolve rapidly. And in fact, just looking at papers that are published these days, rapid evolution is everywhere. Just example after example. Well, why do I make a point of this? What I would like to think is that had Darwin been aware of this, had he understood that evolutionary change could occur rapidly, he would have tried to test his idea of evolution by natural selection. Had he known that populations could change quickly, he would have done the experiments to test his ideas. But of course, he didn't know that, and so he never tried that. And it wasn't until very recently that the idea that you could actually study natural selection in the field experimentally was born. And really the first person who did this was the work on guppies that is justly famous, started by John Endler and taken over by David Resnick, showing that if you put a guppy, well, showing that guppies in nature, when they occur with predators, are bland in color, but you take the predators away and the males get very colorful, presumably because females prefer colorful males for reasons that aren't clear. And so Endler did an experiment where he simply took guppies from a place where there were predators, put them in a little pond where there weren't predators, and very quickly they evolved great differences. They became much more colorful over the course of very few generations. So this work was done in the 1980s. It was done just as I was in an undergraduate and starting graduate school, and it made a very big impression on me. And so when I finished my work here at Berkeley, so now I'm going to get kind of autobiographical. I hope you don't mind this, but my research over 25, 30 years has basically had two thrusts. One is studying adaptive radiation in anolis lizards at the macroevolutionary level. And that was my PhD thesis, and it has continued to this day. And that's what I talked about in Herc Group on Monday. The second thrust of my research has been looking at the mechanisms of evolution in anoles. And this is a project that had its conception just as I was leaving here in 1989. And my talk today is going to be the story of how a research program has developed in ways I never would have been predicted or intended over 25 years. And so this is a somewhat unusual structure for a talk. It's very chronological. First we did this, first we did that. And that's the underlying theme, is to show you, what it's like to have a research program and how it changes through time. I hope that's a useful thing to see, those of you who are starting out in research, just to see the quirks and contingent events that can shape a research program. So having said that, I'm going to talk about the research that I've done on experimental field studies of evolution in the Bahamas. And this is, I call it a play in 5 acts. We may only get to 4 of those acts. We're going to start with experimental studies of adaptation to different environments, then look at the role of interspecific competition on evolutionary divergence, then look at the role of predation, then take a detour to talk about the role of founder effects, and then if there's time, and there may not be, get back to looking at more studies on the role of predation. Now at the outset, I need to recognize the cast of characters. This has been an intensely collaborative project for its entire time. It started during a postdoc with Tom Shaner at UC Davis, the renowned ecologist. Dave Spiller has worked with Shaner ever since he got his PhD at Davis, and so a long-term research associate at Davis. In the more recent years, 2 other people have joined us, Manuel Leal and Jason Colby. They were both students in my lab. They're now professors in their own right. Manuel at the University of Missouri, Jason at the University of Rhode Island, and we continue to collaborate on this project. So having said that, let's start with the first part, studies of adaptation to different environments. Now this work got its genesis in a paper that Tom Shaner wrote in 1983, in which Shaner and his then wife, Amy Shaner, looked at very small islands in the Bahamas, and they saw that there were no lizards on these islands. And so they assumed that the islands were too small to support lizard populations. And so they decided to study the process of extinction by placing lizard populations on these tiny islands and watching them go extinct. And to learn something about the process of extinction, which even to this day we need to know more about. And so let me just show you where this occurred. This is the study organism, the brown anole, Anolis sagrii, native to Cuba, but also native to the Bahamas. Here's where the work was done, in the middle of the Exuma Keys, right here in the middle of the Bahamas. And the work was done on very small islands. Here's some of the islands that the Shaners looked at. Here's a map just giving you a scale. So this is a kilometer this far. And we're talking about these quite small islands. Tiny islands. Some of the islands are the size of this room. So very small islands. They do not naturally have anoles on them. And so the Shaners put either 5 or 10 lizards on the islands and monitored them over several years. And what they found, to their surprise, is that this is the time to extinction of a population in years. And what you see is that above this size island, the populations didn't go extinct. Now, if you can read this axis, this is 10 to the first in meters, so 10 square meters, which is maybe about this big. Any island larger than that, you put lizards on them, the populations persist. And so, in fact, they didn't study extinction. They found that, in fact, these islands can support lizard populations if the lizards get there. And why the lizards aren't on those islands, we'll come back to that later. So, this is all prelude to my own work. I read this work when I was a graduate student here. I had the opportunity to meet Tom Shaner at a meeting in 1988 or 1999. I was thinking of where do I go for my work on adaptive radiation? And I spoke with Shaner. I said, has it ever occurred to you that what you've actually done is done an experiment on lizard evolution? You've taken lizards all from one population, you put them on a variety of islands that vary in their structure, and we know in a macroevolutionary perspective how they ought to adapt to the different environments. Have you ever thought of going back and looking at them? And Shaner said to me what I could only have hoped in my wildest dreams he would say, but never thought he did. He said, oh, what a great idea. How would you like to come do a postdoc and do that? It's like, yes! And so I managed to get funding, and so that was my postdoc at UC Davis. So I guess the lesson of this is it never hurts to ask. And, you know, it's worth talking to people. And if you a lesson that Harry Green taught me, my major professor, if you treat people like a colleague, they will treat you like a colleague. So these are the islands in the Bahamas. They vary in vegetation structure. Some of them have relatively large bushes and trees and so on. Others very scrubby, very low-lying, narrow vegetation. Now, from the work on the anole radiation at the macroevolutionary level, we know how lizards adapt specifically to using differences in surfaces. On broad surfaces, they tend to have long legs, whereas when they use narrow surfaces, they have short legs. And sure enough, these islands did vary in their structure, and so we made the prediction on the scrubbier islands, we expected them to have evolved shorter legs than on the islands with trees, although never trees this big. But we expected to see a difference in limb length that may have evolved over the course of about 15 years. Now, just to show you what field work was like then, and what a dork I was back then, this is me measuring the lizards. And so we would take our little plastic chair, and this is a little monoscope, and I'm counting the number of scales on the lizard's toe. We'd catch the lizard, we'd measure it, I'd stretch out their legs, and then we'd let them go. And I point this out just to show you later on, you'll see how our abilities have changed through time. So in any case, we went back to the islands about 15 years since the populations had been established, and here's what we found, that there was in fact a significant relationship between how broad the surface they used. These are different island averages. So the average perch use and how long their legs are. And this is statistically significant, and it indicates that over the course of 15 years, the islands the populations have adapted to their different circumstances exactly in the way predicted. That as on the narrow surfaces, which is clearly the derived state, they've evolved shorter legs. I won't go into the details, but this was a study. This was seemingly demonstration of rapid evolution over a very short period, and for fluky reasons, it got an incredible amount of attention. For one thing, Nature putting out a press release saying this might be the most important study since Darwin visited the Galapagos, which was a little over the top. Anyway, so it got a lot of attention. I would give talks on this, and there was always, in these talks, there was always someone in the audience, usually a pesky botanist. What about phenotypic plasticity? And you know, you give a plant more water, it grows better, and so on. But these aren't plants; these are lizards, and this is. It sounded ridiculous. And a lizard growing up on a different size surface could grow different length legs. And so I got tired of getting that question, so I looked into the literature on lizard limb length determinants. And that was easy to do because there was no such literature. However, there was a literature on vertebrate limb growth, mostly on mice and other lab animals, and what affects how the limbs develop. And I have to say, and I'm being recorded, so this makes me a little nervous, that this is the craziest literature I've ever seen in terms of the studies that get done. Once there was someone in the audience who did this sort of work, and that wasn't so good. But, so it's studies like this. You put a growing mouse on a treadmill and make it run, a wheel I mean, and make it run 10 hours a day, or some many hours, and then you have your control group that doesn't get a wheel. And at the end, you see if their legs are different. Or you put a rat in a bathtub and make it tread water for hours on end every day, and then the controls don't do that. Or you put a chicken on a treadmill. Now, I know that's not a chicken, but it turns out there are no pictures on the internet of chickens on treadmills. That I could find. And again, you get the chicken to run and the control group. And the answer from these studies is it does matter. Those animals do differ in their limbs, in the bones, but the difference is in the thickness of the bones, not in the length. The length is, of course, what we're interested in. It turns out, like weightlifters. Weightlifters have much thicker bones because you put more stress on the bones, they deposit more calcium. And the same thing sort of happens. But it doesn't make the limb grow longer. And there is one exception in the literature back then, and that came from humans, from a study in the 1950s, and it was actually on professional tennis players. And the cool thing about this was that you could use a professional tennis player as his own control and compare his hitting arm with his non-hitting arm. And if you think about it, professional tennis players have been smacking tennis balls throughout their growth years, since they were little boys. It was on males. And so they had this very different environment. And so the research measured their limbs, and their serving arm was longer. And these were x-rays, so it wasn't the arm was hanging lower, it was the bone was longer. And so there was at least some evidence that environment could affect limb length. Now, of course, it's a very long way from professional tennis players to lizards on different surfaces. And so whether this was relevant or not, we didn't know. So we decided to do an experiment. And so we got a lot of baby brown anoles, and we grew them at the St. Louis Zoo. We got them when they were little tiny anoles. And on the one group, we put them on broad surfaces, 2x4s. Now, this is what brown anoles like to do. They like to sit like that, and this is what they like. This is a very happy lizard. Not so happy lizard. This is the other group on very narrow surfaces. And so we grew them over the course of several months, and as they approached adult size, we measured how long their legs were. And what we found is that, well, first, males had longer legs than females. This is adjusted for size, but we know that. It's a sexually dimorphic trait. However, there was also an effect of diameter. In both males and females, they grew longer legs on broad surfaces. In other words, leg length is a phenotypically plastic result. Well, I almost fell off my chair when I got this result. We actually did the experiment twice just to make sure, and we got the same result, and then other people have done similar things. So it seems to be real, that there is some phenotypic plasticity in anole limb length. Now, the logical thing for us to do would have been to go back and catch some animals and do a common garden to see if this explained the differences we saw in the field, in our populations. But the problem was that the populations were very small, and we didn't really want to take a whole bunch of individuals out. So we kept on putting it off, and it turns out we waited too long, and the populations have gone away. And so we can't do that anymore, unfortunately. However, if you look at the amount of difference between the broad surface and the narrow surface, this is the size difference in limb length. And this is about as big a difference as you can get in environments between basically a flat surface and a very, very narrow surface, certainly greater than the difference that the populations were experiencing. You get this much difference in leg length. And compare that to the amount of variation among natural populations or among our experimental populations. And so I realize this isn't the most compelling evidence, but I would argue that since that the natural variation or the experimental variation far surpasses the amount that you see in our that we experimentally induced. And so probably there is a genetic basis for those differences. But clearly this is not as great evidence as we would like. Hopefully, with all the work going on limb development in vertebrates, including lizards, we can actually begin to get at the genetic basis of this very soon. But that's as far as I can take it at this moment. So let's move to Act 2, and that is the role of interspecific competition in driving niche divergence and then adaptation to different niches. This is, of course, the classic idea of character displacement, competition leading to character displacement. And so we decided to do an experiment on this. To do that, we moved from here, where we've been studying on Staniel Key, a little bit to the south, to the island of Great Exuma, and in particular to small islands around Exuma. Now we have to introduce a second character, a second cast member on the lizard side. This is the green anole, Anolis smaragdinus, a close relative of the American Anolis carolinensis. This is a more arboreal species than the brown anole. These are both very common throughout the Bahamas. And so we had these two species, and we did a similar sort of experiment on small islands. But this time we found 15 small islands that didn't have any lizards, and we grouped them into sets of 3 very similar islands. And within those 3 islands, we randomly assigned 1 island to get brown anoles, 1 island to get green anoles, and 1 island to get both. So we have 3 treatments, and then replicated those treatments over 5 blocks. And so the question was, do the species interact? And if so, do they shift their habitat use? And if so, do they then adapt to using different habitats? And so the first question specifically was, can Anolis sagari, the brown anole, survive on these small islands? Can the green anole survive? And then, do they interact? So here is the data for the brown anole. We put 5 lizards on the island. 1 year later, the populations have at least doubled in size, and several of them have truly exploded in numbers. 3 years after the introduction, 9 out of the 10 islands are very well established. Oddly, something happened here. It was down to 1 lizard, but this island actually recovered. So the brown anole does just fine. What about the green anole? Well, it's a bit of a different story. 1 year in, 8 out of the 10 populations have at least doubled in size. But 3 years after the introduction, the story is a little different. 4 populations have gone extinct. 2 other populations are down to 1 individual, and in both cases that individual is a male, so that population isn't looking good. And really only 2 of them are doing very well. So the green anoles don't seem to do very well on these small islands, and maybe that explains why you don't find them there. What about, is there evidence for interaction between the 2 species? Now, as you'll recall, there were 5 blocks, and in each block there was an island of green anoles alone, and 1 with green anoles and brown anoles. And so what we see here is the density of the green anoles on those 2 treatments. And particularly if we focus on the middle of the experiment before populations crash, we see that in all 5 cases, the red line, where the green anole was allopatric, is higher, has a greater density than the green anole in sympatry with the brown anole. Put another way, the brown anole has a negative effect on green anoles. You have brown anoles, you're going to have fewer green anoles. They have a negative interaction, which is probably interspecific competition for resources. So I think we can explain why we don't see green anoles on these small islands. Number one, even in the absence of brown anoles, they went extinct on a few, and then the brown anoles make it even that much worse. But why don't we see brown anoles on these islands? This is now 2 experiments in different places that show if you put brown anoles on these small islands where they don't occur, the populations do great. Why aren't they there naturally? Well, that suggests the idea that something occasional, something catastrophic, affects their ability to persist on these populations. And of course, when we're talking about the Bahamas and talking about catastrophes, we're talking about hurricanes. Now, this research program, that going back to the days that Tom Shaner started working in the Bahamas, Shaner had been studying lizards and butterflies and other things for years, and he basically had the year-to-year dynamics down, but but what happened, unfortunately to Shaner, is just as he started working, right around here, the hurricane frequency really dropped. And so he had never experienced a hurricane in any of his studies. And so he had postulated in the original paper that the reason brown anoles weren't on these islands was due to hurricanes, but he'd never had a chance to test that idea. Well, in 1995, that chance finally came. This is Hurricane Lily, which is churning its way across Cuba and headed right towards our study site. And we were very excited about this. Here's a chance to test this idea. What effect would it have? So we were excited, but we also there was a downside, and that was that we were on the islands at the time. So Lily goes across Cuba, headed straight towards it, but then it takes a hard turn to the east, and so it was going to miss us. Too bad. But then it turns right back. And so we just followed it. We listened to the BBC, the Bahamas Broadcast Corporation. Every 3 hours they would give us an update, and the banks distributed a little map just like this, and we could plot the hurricane going closer and straight at us, and sure enough, it was a bullseye hit. The eye of the hurricane went directly over us. And so the effects of the hurricane were dramatic. There were many trees that were just toppled over. We counted 17 yachts that were destroyed, very sadly for those people. And it had a huge effect. By incredible good fortune, we had just finished our surveys for the year. And so we were in the almost unique perspective of being able to say, 3 days ago the island was like this. After the hurricane, it's like this. And so one thing I didn't tell you, here's our study site in Georgetown. Our islands, our study islands, were distributed on both sides of Georgetown. Now, that really shouldn't matter. Hurricanes do slow down as they go over land, but Great Exuma is only 1 mile wide, and that's not enough to make much of a difference. So we started off by going to the far side, the northeast side. And what we found when we resurveyed the lizard populations, well, number one, you go to those islands, clearly something had happened. Leaves were knocked off trees, branches were broken, you knew something had happened. And we resurveyed the lizard populations, and they were down from 40% or a third from what they'd been just a few days before. So clearly there was an effect of the hurricane, but also just as clearly that effect was quantitative. It wasn't a state change, if you will. We then went to the near side islands on the southwest. This was my favorite island. This is an island that was the one island that the green anoles had really done well. There were 125 of them, just everywhere you took a step, green anoles jumping everywhere. Delightful island. So we get to this island, we didn't even recognize it. And the reason is that all the vegetation, except the stuff deep in the cracks, was all gone. And the other islands were like this as well. This was a big tree with a big 10-foot wide canopy. Another one, they were both snapped in half, all the vegetation gone. Here's yet another island. This had been an enormous bush, washed away. The island was like a bomb had hit them. When we went to census the lizard populations, that turned out to be very easy. The lizards were all gone. And so we could see, in fact, this is why, as Shaner had predicted, the islands have no lizards. Because every once in a while, a hurricane comes along and clears them off, and they just don't disperse that quickly so that most of those islands, the small islands, remain empty. But the last question is, how did the hurricane actually accomplish this? Because as I said, there's a difference in the two sets of islands, and it shouldn't be the wind speed should be the same. Well, there was one clue, and I know many of you have taken marine biology, and so you recognize that this is a starfish. I guess we call them sea stars now. And you know that sea stars don't occur on the top of islands. What this indicates is the islands were underwater. And we now know this from Katrina and other things. There was a big storm surge that hit the island with great magnitude, washed away all the bushes and all the vegetation, washed away the lizards, but then the storm surge hits the big island of Great Exuma, that stops the surge, and there isn't enough open water on the other side to build up another one, and that's why the far side did all right. So that, unfortunately, ended our experiment. So it goes. That leads us to Act 3, studies of predation. And in particular, what role does predation play in anole habitat use and adaptation? So once again, we move this time up to Abaco in the north of the Bahamas, where we will remain for the rest of this talk. And finally, our last actor in this play is this lizard here, this charming lizard called the curly-tailed lizard, Myocephalus carinatus. Now, it may look friendly and adorable, but in fact, it is a voracious predator. And it is known to eat brown anoles. But what we didn't know is whether this was something that happened very rarely and had no real consequence at the population level, or whether this was a huge factor in affecting their populations and potentially their adaptation. And so what we did in the same sort of way, we got 12 small islands. Now this time the islands were a little larger. They had brown anoles naturally. And on half of these islands, we randomly decided to put half of the islands, we'd put the curly tails on them, and the other half we wouldn't. I do need to point out, the curly tails occur on large islands right or in the same vicinity. We're not bringing something from far away, we're bringing something from 100 yards away, more or less. So we're mimicking the natural process of colonization. So that was the experiment, and I want to show you a little film clip of how it went. I had the good fortune to work with the Howard Hughes Medical Institute to make a video on some of these research, and they did a fabulous job in actually explaining this better than I can. So I'm going to play the little clip. The one thing I have to tell you is we argued about this a lot, but they keep on referring to Losos and his team or something like that. This is not my team, these are my collaborators, and we argued about how that had to be phrased, and I didn't entirely win. So let's see if you can hear this. Oh no. Well, I don't know why. I should have tried this already. It was on a minute ago. It's still, oh, did it?
Come unplugged.
All right, well, I'll have to narrate. Losos catches the lizards. This is Jason Colby, my colleague. This is the bad guy, the curly-tailed lizard, looking around. There he is. And Losos wanted to check what effect the curly-tails would have on the lizard population. There I am expounding. This is the bad boy of our experiments. It eats brown anoles. And will there be any selection on the brown anoles? That's essentially what I'm saying. Lizards running away, the brown anoles chase them. Our expectation is that the lizards that have longer legs and can run faster will be favored. And so selection should favor longer legs. That's what I said. So here's what we found. This is the population density. The open circles are the controls. The closed circles are the islands with curly-tailed lizards. And what you see is very quickly there was a 50% drop in lizard abundance in the presence of curly-tails. And what happens is these are the babies being born, and what you can see is the babies are getting eaten. And so the population doesn't go up so big, and then it comes down, and that persists throughout the experiment. Moreover, this is perch height, and what you see that these anoles are no dummies, that even though they have not seen curly tails in their, for generations on this island, as soon as you put them on there, they shift up higher into the bushes. I forgot to say the curly tails are almost entirely terrestrial. And so the lizards are moving up into the bushes to get away. Now we don't know why they go higher up over the years, but that trend continues. So a very clear population level effect. And our prediction is that this should lead to a change in limb length. Unfortunately, this is what happens. Hurricane Floyd, 1999. Here's our study site. Islands are wiped out. Bummer. However, we decide to do it again. You have to wait a few years for the in this case, the lizard populations, the brown anoles, weren't wiped out. They were knocked back, and the vegetation was damaged, but they recovered over a few years. The curly tails were eliminated from the islands. So we do the experiment again getting a bunch of islands, randomly assigning them, but this time we did something different. And before introducing the curly tails, we captured every brown anole on the island, measured how long its legs are, and injected them with a non-toxic rubbery substance that was essentially the same as a bird band. So here's orange on this leg and green in here. And so we could individually identify the animals. We knew how long their legs were and how big they were. And then later on, we would go back and see who survived and who didn't, and whether limb length affected survival. And here's another video, but since I don't talk, yeah, we'll skip the video. Is it? Too bad. I should have tested that. Oh well, so it goes. I'll show you the results anyway. Again, there was a very rapid change in perch height. It's expressed differently. This is the number of lizards seen on the ground. And within 6 weeks, there's already a difference. In the presence of the curly tails, they're on the ground less. And 6 months into the experiment, there's a threefold difference in how often you find them in the ground. So in the presence of the curly tails, they're on the ground much less. So again, the same habitat effect. So we went back, we recaptured all the lizards, and we measured natural selection. And so basically, natural selection in this case is just asking, are the leg lengths of the survivors longer or short, different than the leg lengths of those that didn't survive? And so we measured what's called a selection gradient. And so a selection gradient, if they're a high value, means that the survivors have longer legs than those lizards that didn't make it, indicative of selection for long legs. A negative value would be selection for short legs. A value of 0 would be no selection, that the survivors and non-survivors don't differ in leg length. And so we calculated the selection gradient across all of our experimental islands, all 12. And what we found was a significant difference. There is selection in the experimental islands for longer legs, exactly as we predicted, because we know longer-legged lizards can run faster and presumably escape better. whereas no net selection in the absence of curly tails. Well, this is very exciting that by putting on a predator, we've changed the selection milieu. So selection has changed. However, we also in this experiment, well, this is from the previous experiment, we know that they're moving up into the trees, the bushes, to get away from the curly-tailed lizards. Well, what that means is they get high into the bushes, they are using narrow surfaces. These are the curly-tail islands. They're using much narrower surfaces in the presence of curly tails. And we know how lizards adapt to narrow surfaces. They evolve shorter legs. So our prediction was over time, once the lizards have really moved up into the bushes, selection should turn the opposite way and start favoring short-legged lizards. And sure enough, that was what we found, much to our surprise. In the second 6-month period, comparing those that had been alive in November and the ones alive in November, whether they survived to May, now selection was favoring short legs, a reversal in selection over a 6-month period. Well, this was, of course, very exciting, but moving forward, it's not like it's going to go back and forth, because now they're stuck up in the bushes because the curly tails are there. So year after year, we should now see selection for short legs. And over time, the prediction would be that they should start evolving to look like twiganoles. So what happened? What happened? Anyone?
Hurricane.
No! 2 hurricanes. Hurricane Francis, September 3rd. Hurricane Jean, 3 weeks later. Islands got wiped out. And so once again, I never knew I'd be a hurricane expert. And we now have learned a lot about what hurricanes do to ecosystems. All right, so however, there was a silver lining to these hurricanes. In this case, it cleared off a bunch of islands and allowed us to do an experiment that I wanted to do for a long time. And it had to do with the founder effect. Now, you all are probably familiar with the idea made famous by Ernst Mayr that founder effects, when a population is created by a very small number of individuals, that may lead to great evolutionary divergence just by random sampling. That if you can imagine, if any 2 of us were stranded on an island, a male and a female, our particular traits would lead to a new population quite different from the population as a whole. Mayr argued that this could be important for genetic divergence, perhaps even speciation. Well, this has been a controversial idea. I would say in recent years it's fallen out of favor. But this idea has really, it's been tested in the lab with Drosophila, but really there's very little data from nature, certainly not of an experimental nature. And so we decided to do a founder effect experiment where we would just create replicate founder-created islands and just see how they diverge and see how that would interact with selective factors. And so what we did is we put a male and a female on each of 7 islands, all drawn from the same nearby island. We asked, would the population survive? Would we see a founder effect? If so, would we see it in the genetics or the morphology? And would it persist through time, particularly in the face of directional selection towards, we expected shorter legs on these scrubby islands. And so here's what we did. Here's the source island in the background. This is Tom Shaner, my colleague. Here's the scrubby island. So we took lizards from here, put them on here, a male and a female, 7 different islands. Here's just a look at a few of those islands. And first, did the population survive? Well, they did. We got fortunate that the first year was a very good year, and the populations all grew up very quickly, up to as many as 40 individuals 2 years later, and then they kind of wobble around. But all 7 populations did just fine. Well, let's look at genetic variation. To do that, we look at microsatellites. What we have here is a principal coordinates analysis of the genetic variation, distilling it down to 2 axes. The black dots are the the source population. This is the average of the forested population. The colored dots are the individuals we put on the different islands. So here's and we just randomly assign them. So here's the 2 blue ones, both up here. Here's the 2 purple ones, and so on. And if you just look at the average of those individuals, you can see we created a founder effect that these 2 islands, these 2 individuals are quite different from the mean, and these 2 vary over here. And so then we followed the population through time to see if this difference induced by the founder effect persisted, and sure enough it did. This is 1 year later, and they're still quite different, and so on. And this is 4 years later, and the axis has changed a little bit, but you can see that these differences that we created, this genetic founder effect has persisted over the course of a few years. Now, we also looked at the morphology, and to do that we x-rayed them. No more me sitting in a plastic chair. And x-rays are really cool. And I don't have time to show you other than here's a cannibal with a little baby in his stomach and a snail. You can see the gravid ones, there's an egg there and so on. Here's another place where something is snipping the legs off these, and it's lots of fun. We also scan them to measure their toe pads, that's Jason Colby. We also measured their dewlaps with this contraption, Manuel Leal. I'm not going to talk about this, but it's a very accurate way of measuring it. Was there a founder effect in morphology? Well, this is the founders plus the F1, and what you see, here's the source, and there are great differences. The individuals on this island have much longer legs than the source, and this island has much shorter legs. So the founder effect has created, one generation later, great differences in morphology. So there was an initial founder effect in morphology. Would it persist through time? In the face of adaptation, because they're on these scrubby islands, we expect selection to be favoring shorter limbs. Would it just wipe away the evidence of the founder effect, which I think many people would predict that it would? Well, the answer is yes and no. There is a clear predicted decrease in leg length across all 7 islands, and that shows the year effect on limb length in Ancova. But there's also an island effect, that the islands in general, the initial differences have been retained statistically. The initially shortest legged ones have stayed short, the longer legged ones tend to be long, and so forth. And so this indicates that over several years, the founder effect in morphology, even in the presence of strong selection, you can still see its imprint. Well, this was of course only 3 years of a founder effect, but it does show that at least in the short term, founder effects can have a long-lasting impact. You could criticize us for not going longer, but this is the data we have. It was persisted for at least 2 more years. I'm going to skip this and that and just show you that, as you might have expected, another hurricane. This is Irene, which you may remember, but you've probably forgotten about Irene because of Sandy. And both of them went over our study sites, and unfortunately, only 2 of our islands remain. So, of course, we were hoping for more, but that's what we got. We may redo this founder effect experiment, or we may not, but we have redone the predation experiment, and I'm running out of time. I just want to give you one last result. We're getting the same result, a difference in population size in the presence of curly tails, difference in perch height. But what we did differently is that we started filming the behavior of these lizards in great detail. And when I say we, I really mean Manuel Leal, my colleague, and his cast of extremely hardworking undergraduates and graduate students who just videotaped these lizards for 30 to 40, I think 30 minutes per lizard on the islands. And the most interesting result, I'm just going to skip through this, is that they show a difference in how much they bob their head. Lizards kind of go like this. And on the non-curly tail islands, the lizards have a high amplitude bob. But on the curly tail islands, their bobbing is much smaller. Well, you can actually calculate how far a signal can be seen based on, if I go like this, the people in the back can't see me moving my finger probably, but those up here can. And similarly, a lizard, the bigger the bob, the farther away you can see it. And so this difference in amplitude suggests that on the non-curly tail islands, a displaying male can be seen this far away, but on a curly tail island, the area that males can be seen is substantially smaller. And so the implication is that the presence of curly tails may have an effect on the social structure of these lizards, that it may in some way perhaps affect their ability to hold territories, which may change selection pressures perhaps on sexual dimorphism or in other characteristics. We don't know yet, but it certainly suggests another aspect in which predators can affect the evolution of these lizards. And this is ongoing work that we hope that the hurricanes will stay away and that we can get completion to one of these studies, or at least a longer time distance, but the work continues. And with that, I'd like to thank you for your attention. It's great to be back.
Wonderful. We've got time for questions. If someone can hit the lights, please.
Have you considered, if a hurricane is coming, quickly harvesting all the lizards so you don't lose them all?
The problem is, the first hurricane we were at was really interesting because there have been a large number of false alarms. The hurricane's coming to it and then veering away. And so the people in the town we were staying were completely ignoring it. which was driving me crazy because it was coming right at us, and we were staying in a very exposed place. And so the problem is that it's only a very short period of time.
Okay.
The other idea people have suggested is maybe we should put some cameras on the islands and film what the lizards are doing.
Cameras.
There are all kinds of things in theory it would be fascinating to find out about. Yes?
Yeah, just sort of a follow-up to that. With those marked lizards, do any of them end up showing up in other islands after?
Very good question. We've never found one either on another island or even anywhere else. Nonetheless, the study I showed you with the microsatellites seemed to indicate a much higher level of gene flow between islands than we would have predicted. However, those particular islands are right next to each other. I mean, literally some of them are separated by a few meters, so they're very close together. So, but we clearly need a more detailed population genetic study. Maybe they do get back and forth more than we would have anticipated. But no, we never saw them. And I have to tell you, after that first hurricane, when we were right there, we went back out, and the water was just full of vegetation that had been thrown into the water. And we actually got one of Rosie's spiders that was only found on the big island and showed up for the first and only time on one of our little islands and lasted for a few months and then petered out. But stuff does move around in hurricanes. Yes?
In your founder effect experiments, in those islands that started out by chance with longer limbs, did you see an effect of genetic load there, of more mortality?
So the question was there more mortality in the founder effect experiment on the islands with longer legs? But we didn't mark individuals, so we'd only know it in terms of population.
Population size.
Yes, and I don't think so. I don't think so. I'd have to go back and look. It's an interesting question. Correct.
So Jonathan, on one hand you have this evidence for repeated evolution of ecomorphs on the islands that sort of may have perhaps involved in speciation or not, but certainly with character displacement. And you've got this beautiful sort of microevolution stuff with fluctuating natural selection. And I wonder in this case and the sort of work that grants to, how much of this is sort of, even though it's demonstrating natural selection, is sort of evolutionary noise? And under what circumstances does this create sort of durable change that goes beyond the noise level?
Well, that's a very interesting question. One of the great things that the grants have shown us is that natural selection can fluctuate much more than anyone had predicted from one year to the next. And certainly I think we're seeing that in this case. And in fact, we have a slight bit of evidence that the hurricanes may actually be selecting for long-legged lizards, that they may actually be able to hang on better in hurricanes. Data sort of suggests that. But that suggests that similarly on these islands it might be going backwards and forwards. And I think that that's something that only recently has gotten the attention of evolutionary biologists, is that you sort of think of directional selection generation after generation like a lab experiment. It's really not like that at all, I think, in many cases. And so finding out the long-term pattern from the short-term noise, I don't think we know the answer. I think in the case of these islands it probably is these fluctuations, or if we could let it go long enough, we'd find out. But I think that's a very important perspective, and it certainly shows how important these long-term studies are. But would these produce twig anoles? If the hurricanes would leave them alone, would they end up twig anoles in some number of generations? That would be fascinating. I'm going to guess no, but I don't know. And it's surprising, even in anoles, which have been so well studied, no one has looked at this sort of question. Yeah, the number of natural selection studies is very small, and it's such a great system for doing this that I hope we can get at that.
Well, you're not done yet.
Well, a few more. Yes.
So, sort of following up on that, I guess I felt sorry for the curly-tailed lizard. You know, it's sort of like Jaws in the movie. It must be evolving as well. What do you know about the evolution that's taking place there, behaviorally even, or?
It's a very good question, and initially, actually, the first experiment we did, we were going to study the curly tails and not the anoles. That was the plan. And there are two problems with that, and one, the biggest problem is that they're really hard to measure. When we measured them then, we were measuring them by hand, and they were live animals, and they thrash around and they don't lay flat.
Yeah,
We
I
couldn't
see
get
that
good.
that chair would not be stable enough. to
Exactly.
deal with it.
I'm the ear and they're wrestling around, and now with our X-ray machine, they're too big for the X-ray machine, and it would be really fascinating. Now, one thing I will say is that the populations are quite small, and so certainly there's got to be a lot of fluctuation, but we don't really at this point have the answer. But it would be very fascinating. They do eat just about anything. So
Okay.
it's. I mean, they eat, sometimes there are these fruits, and they drop, fruit drops, and there they are eating these little mayapple things. So they'll eat just about anything. It would be fascinating. I don't know. Sure.
Sort of related to that question, is there potential for observational bias on some of these studies where you might have expectations before you initiate the measurements? How do you avoid situations like that?
Well, that's, I have to say, the great thing about the X-rays is that they're very repeatable. In the old days, I mean, I tried very hard measuring them multiple times. The one advantage is that I would bring them back into the lab. Every day we'd go out to as many islands as we could and capture them, and I would measure them without actually knowing which lizard came from which island. And I would measure them several times. But that is a very real concern. I love my X-ray machine because you have the record of it, and you can measure stuff more accurately, and you can go back later. And but that is a concern that had worried me.
Jonathan, what can you say about the genetics of limb length? So in these repeated experiments, can you use that as a way of getting at the genetic basis of what?
Well, right now we can't say anything, but you know, there are people studying the genetics of brown and leg length, and they're making progress, and we're really hopeful with all the great techniques that soon we can, I mean, it'd be a perfect, there's such great opportunities, and we haven't gotten there yet, but, and everyone else, I think that in 5 years, 10 years, we'll actually begin to able to look at this and get at that. So, because, you know, the vertebrate limb is a model for development, and just trying to apply that to these guys. So that's where one of the things we're hoping to
I mean, it's nice with the repeated examples that you might be able to look for parallel changes.
That's exactly it. And there, a number of different people are doing that, and I get to collaborate with some of them, and my job is to catch the lizards and say, here you go, and have at it. So I hope we can get at that.
Yeah.
Yes?
So going back to your first hurricane, with your Leeward Islands that experienced 40–50% population loss, do you have any sense whether was any selection associated with that? Or, you know, is it just a random decimation of the population, or is it possible for the hurricanes themselves to impose selection?
Well, so on that experiment, so we didn't have individuals, and so the question would there be a shift in the population mean? And I'm embarrassed to say I don't know that we ever looked at that. This is a while ago now. I don't know the answer to that one, but I will tell you that after Hurricane Irene on the Founder Effect Islands, most of the population survived that, and 6 out of 7, the legs got longer that year. They've been getting shorter, they got longer. And one of our teams said, hurricane-based selection. I said, no way, that's ridiculous. And then the next year, another hurricane, the 2 surviving islands, they got longer again. Could there, and so the idea is maybe the longer leg guys can hang on better. We actually put some, we actually put some on sticks and pulled them off with the Pasola scale, and the longer legged ones maybe can hang on better. We have this experiment, we've got permission to do it, to put them in a wind tunnel. So maybe, I still don't really believe it, but maybe. So we'll see that.
All right, well, I want to remind everyone we're having a reception tonight for those who would like to chat more with Jonathan. I know his schedule was very full and not everybody could get on it who wanted to, so please come tonight, and let's thank Jonathan for a great talk.
Thank you.